My name is Lizzie.
And I'm Mateus.
And this is a stop motion of the process of meiosis. To introduce this process, understanding the cell cycle is very important. The cell cycle is a series of steps during which the cell grows and divides. The cell theory states that the cell is a basic unit of life. Every living thing comes from other cells, and all cells come from other cells. In mitosis or regeneration, the cell is asexually replicated exactly into a daughter cell. In meiosis, or sexual reproduction, the cell creates genetically varied daughter cells. This video will explain the stages of the cycle of meiosis. The first stage, interphase, is the longest phase, preparing for division. In G1, the first step of interphase, the cell grows and copies its organelles. In S, the second stage, the DNA is duplicated in order to prepare for cell division. Finally, during the last step, G2, the cell synthesizes proteins and hormones needed for division. After interphase, prophase 1 begins. In this, chromatin condenses and the nucleolus disappears. The homologous chromosomes, or the similar ones, are stuck together as pairs to perform synapsis. The two homolog chromosomes form at the chiasmata and leave genes in portions of the other chromosomes. This event, called crossing over, is how genetic variation can happen. Also, in prophase 1, centrosomes are copied and migrated to opposite poles. The spindle apparatus forms for metaphase here, and the nuclear envelope breaks down. Following prophase 1 is metaphase 1. In this phase, the tetrads assort in pairs down the equator of the cell, or the metaphase plate. Spindle fibers attach to the centromere of the chromosome in the region, the kinetochore. The centrioles are placed at opposite sides of the cell, fully forming in the spindle fibers. Spindles connected to the kinetochore are named kinetochore microtubules, but some microtubules do not attach. The next phase, anaphase, begins after. In anaphase 1, each duplicated pair of homologous chromosomes will be attached to a microtubule when the kinetochore microtubules shorten. Homologous chromosomes get pulled apart in this shortening and go to opposite poles of the cell, which elongates as microtubules push poles of the cell farther apart. Telophase is after this, which is when the chromosomes uncoil and a nuclear membrane is formed around them. The cell pinches in, which creates cleavage furrows and begins cytokinesis. In cytokinesis, the actin filaments around the cell will tighten. This pinches the cell membrane, causing two different cells to be made. After the split, meiosis 2 can begin.
Meiosis 1 and 2 are very similar, but are different in that meiosis 2 has another cell with half the DNA as meiosis 1. It starts with prophase 2, where the chromosomes condense again, and the nuclear membrane breaks down. The spindle between the centrioles forms again to attach to the chromosomes. In metaphase 2, the centrioles are at the poles of the cell, and the spindle is fully formed. Each chromosome goes into a line in the middle individually on the metaphase plate. In the kinetochore, spindle fibers attach to the centromere. In anaphase 2, the kinetochore microtubules shorten, and sister chromatids, which are attached to a microtubule, split to opposite sides, lengthening the cell. In telophase 2, chromosomes uncoil, and a nuclear membrane is formed, again around them, while the spindle also goes away. The cell pinches in cytokinesis. The actin filaments around the cell will tighten. This pinches in the cell membrane, causing a cleavage furrow. After cytokinesis, you can have spermatogenesis or oogenesis. In oogenesis, the cell produces and develops an egg. In spermatogenesis, the cell produces and develops a sperm cell. Thank you for watching.